finally we get to the last lecture. And here, what I want to do is spend a little time going over what I would call some oddities, some behaviors that are a bit non-standard compared to what we've seen so far, as well as a bit more models, just to give more variety in terms of what we see. Um, so, as I said, so first of all, I'll look at uh, some models that behave a bit differently, and then a few recent models. And to finish, I'll very briefly mention what I have not mentioned, which is a lot. Um, so, in terms of additional considerations and things that go a little weird, let me look at the following problems. So there's one where I'm going to show you a little problem with the computation of R0 using the next generation method. I'll show you a model where a tuberculosis model where the behavior at the origin is hard to deal with. I believe I might have something with non-constant population. I'm not entirely sure that I still do, but I'll definitely also talk about bistable states. Um, so first of all, this issue, so to speak, with the next generation method. And I'll point out this is not an issue with the method itself. It is an issue with if you apply the method without really thinking about what you're doing. And so this is a model for malaria, uh, including transgenic vectors, which myself and collaborators published quite a few years ago. And I'll show you how this R0 business can go a little weird at times. Um, so, as I've already indicated, this is not a problem with the method of next generation matrix, but it's an illustration of why it is that those conditions that we saw in practicum 02, I believe, are sometimes important to check. Now, the model that I'll be considering is the following, and let me get myself out of the way. This is a vector host model, a very classic vector host model in some respects, and quite different in other respects. So let me explain the context. So here we're looking at a malaria problem, although we're thinking about any vector host. But the idea that was underlying this work was that in some contexts, you can try to drive some genetic aspects in mosquito populations, vector populations, but mosquitoes, with without oops, what I need. You can drive some genetic components in a sort of non-Mendelian way. It's a complicated process, but the idea that this model was investigating was one that was considered at one time, where there was a method where you could have transgenic mosquitoes who would have a different behavior with respect to the way malaria developed within them. And so that's an important distinction between some vector host problems and other vector host problems. There are diseases which I mentioned Dutch elm disease in the past at some point, and so there are diseases in which there is the vector is just a vector, whereas like in malaria, the parasite has a cycle that it goes through in the vector and a cycle that it goes through in the host. Okay, and to achieve propagation, the parasite must go through these two cycles. And the problem here, well, I mean, the solution that was being considered was one where the parasite can't really develop within these transgenic mosquitoes. The problem here is that, okay, you're introducing a population of mosquitoes like this with transgenic, so they're the ones here. And to simplify things, we're going to assume that they're immune to malaria. But they reproduce with the non-transgenic, the wild type mosquitoes. And of the offspring that are born from these, like the mating of transgenic and non-transgenic mosquitoes, some of them are resistant, or so are bearing the transgene, others not. Okay, so what happens is we have a vector host model, but where the dynamics of the vector is going to be very complicated, and I'll get back to that. For the humans or the hosts, the dynamics is very simple. Okay, it's a SIR model with disease-induced death, but it's an SIRS, I should say. It's an SIRS model with disease-induced death. But as I said, this is the hard part. Well, hard, the complicated part. It's the mating of the mosquitoes. So the hypotheses we make about the populations of mosquitoes are, first of all, that, as I said, resistant vectors, they're completely immune to the pathogen. So they don't get infected, they don't propagate the infection. Well, I mean, they get infected, but the parasite cannot develop in them, so they don't propagate. And then when you consider the offspring resulting from interbreeding of wild and resistant vectors, some of them are resistant, and whoops, this is weird. And some of the offspring resulting from inbreeding within the resistant types are resistant. But 1 minus P1 of the mix and 1 minus P2 of the mix are not resistant. Okay. And there's also some fitness cost to resistance. So if there's no disease, the wild vectors are better fitted for competition. But if there is disease and they're infected, the wild vectors are less fit for competition. Okay. So the model looked something like this. So this is the population of mosquitoes, vectors. So the SI being the wild type and the T being the transgenic. And this is the population for the dynamics for humans or hosts. We assume that the force of the incidence functions are C1 and they're non-negative and they're zero whenever one of the components is zero. But what happens in terms of the dynamics of the populations? So to begin with, what we're going to do is we're going to assume that there's no disease whatsoever, and we want to know if a population in which there's susceptible, well, wild type and transgenic type mosquitoes can persist. Well, I mean, if a transgenic can become established, that's the more important problem. So you're introducing these vectors that you hope will help control, but if you introduce them and they're unable to subsist in the absence of disease, then it's unlikely that they'll make it. So, and that's where what I was describing, the birth of well, the dynamics of the population of wild type and transgenic mosquitoes is complicated. This is just the population dynamics of those species. There's no disease here. What it's saying is that there's a certain birth rate. So if you take the wild type, there's a certain birth rate that results from mating of wild type and wild type. Then there's the transgenic driven birth, but non-resistant. there's mating, so these are transgenic with transgenic. Then there's mating of wild and transgenic that does not result in a resistant mosquito offspring. And then there's some population dynamics. This is like a logistic type problem. So there's a population dynamics there of the population of wild type. And there's interspecific competition between the wild type and the transgenic type. Okay, and so that's essentially a logistic term, but where there can be interactions between the two species involved. And likewise for transgenic, we have birth of transgenic from the mating of two transgenic. We have birth of transgenic from the mating of a transgenic and a wild type. And we have natural death as well as competition with the wild type in this case. Now, I'm not going to go into the details of the model. Okay, so we can define a fitness for the wild type and a fitness for the transgenic. And what's important is that we find two boundary equilibrias. So there's one where there's no vectors of any type. Okay, both the wild type and the transgenic are zero. There's one which is the fitness of wild type and zero. And then there's a coexistence equilibrium where the two species are present. If there's no loss of resistance in the offspring, then there's another equilibrium which has only transgenic. Okay, that's a boundary. So there's two boundary, there's two boundary equilibria. There's one which is the origin, and there's one that is in the interior of the quadrant. So what we have is a situation like this, which we can summarize in terms of the relative fitnesses of the transgenic and wild type mosquitoes. And depending on where we are in this parameter space, we have either the wild type unstable and transgenic locally stable. We have potentially 3 here. We have 2 here, a single equilibrium here that is globally stable, 1 unstable and 1 globally stable. What's important to keep in mind here is that this is the model that prevails in the absence of infection. Okay. So so there's no I is equal to 0 here. So when I'm looking for the disease-free equilibrium, I end up with the same, problem essentially, okay? Because this is I equals 0. So that means the system has up to 4 disease-free equilibrium. Okay, because each of those corresponds to a situation where I equals 0, so they can all coexist. And we were trying to use the method of Van den Driesch and Whatmore to characterize this. And when we look at E0, so the equilibrium with extinction of both the wild type and transgenic mosquitoes, what we found was systematically that R0 equals 0, which makes no sense. And the problem here is with the assumption A5 in the paper of Van den Driesch and Whatmore, because what you find when you consider this assumption, you compute the Jacobian of the system in the wild type and transgenic mosquito and evaluate at E0, the extinction equilibrium, then you get a positive eigenvalue and another eigenvalue. So that means that equilibrium in the absence of disease is always unstable. So you cannot satisfy A5, which means that you cannot use the theorem of Van den Driesch and Whatmore that we saw earlier. And that is why things go wrong. Okay, so most of the time, I will point out, most of the time that theorem is going to be applicable. But there are instances where it is you're not going to be able to use the theorem, and that is something to bear in mind. Okay, so it's always, I mean, you've heard that before, it's always worth checking that you can indeed apply a theorem. And the problem with this result is that it works so often that most of the time you don't actually end up checking these assumptions, but they're worth checking altogether. Now let me show you a model of McCluskey and Andrisha. In this case, it's something that I've mentioned in the analysis practicum. What happens when things are a bit messy at the origin? So this is going to be a tuberculosis model, but the recruitment is going to be a little complicated. Okay, so in the model, what they have is that the total population N prime is governed by this system, B of N minus Dn. So there's a per capita death rate D, but the birth function is somewhat complicated. So it satisfies these three properties. So there's a unique N0 such that Bn of 0 equals Dn of 0. Okay. The derivative of b is less than the derivative of b over n whenever n is in, that should be, that's a French open parentheses, in the open left and closed right 0 and 0. And they denote b sub 0, the limit when n goes to 0 plus of b over n, b of n over n. And they suppose that this is greater than d, where b0 is in the extended real line. So it can be equal to infinity. So the model they consider is this very simple model. What's important here is you can see there is disease-induced death here. Recall that when you have TB, you can be latent for many years, and so this is a model that makes a very simple description of this. But what's important here is that the recruitment, that B of N function, is complicated, and furthermore, because there's a delta here, it's not simply B of N minus D times N. There is this additional mortality term that makes the total population non-constant. So what they do is they rewrite the model. They recast in terms of N, E, and I instead of S, E, and I. Then they look at the system in proportions. But the issue, yes, so if you look at this system here, the problem is that there are solutions going to 0, 0, 0. Okay. And so this means this system has this potentially going to 0, which means that, of course, things are a bit more, oops, I should move myself out, okay. This means that there are potential complications here, and they are present, okay. So what they do is they extend the system at n equals 0, and they do that by replacing b of n over n evaluated at 0 by that b0 that they defined earlier, which is the limit when when n goes to 0 plus of b of n over n. Okay. Oops. This. So they find that, okay, the biologically relevant region is this one. And they find two equilibria, one which has everything equal to 0, and another one which has n equals 0 and E and I positive. And remember, these are the proportion of E and I in the population, not the numbers. Okay. And so what happens is when you have this equilibrium there, you're going to zero, but in a controlled manner, okay? Because these quantity E and I, they're positive, so you are going to zero, but the proportion of E and I in the population are fixed along this decrease to zero. Okay, so they find three actually basic reproduction numbers to apply to different places in the system, and they find a variety of equilibria. Okay, so depending on the situation, you can see that there is a variety of unstable here. There are a variety of situations that arise. Okay, depending on the situation. Oh, and I still have this. I thought I had got rid of that. But what they find are global results in terms of the behavior. Okay. So, oops, I should move myself out. So here the important thing that I wanted to highlight with this model is really the fact that when you have to be careful in some instances, most instances models behave nicely, for example, when you go to the disease-free, but in a model like this, where the total population can go to zero, you end up having to make much more fine considerations around the origin. Let me look at, yes, so I did have an SLIRS with non-constant population. So this is a model of, this is an old model from Liu, Levin, and Iwaza in 1986. And this one is going to be interesting because it's going to exhibit periodic solutions. So all the models we've seen so far have been quite tame in their behavior. Here, they consider a model like this, and notice that this is the same assumption we saw before in the model just before. Okay, we suppose there's a stable equilibrium n zero such that n of zero equals dn zero, which is just gets this equal there. So look at the model. So there's this birth rate. This is a force. It's not quite a force of infection. It's written in the form of a function that applies on I, whereas if we were looking at a force of infection, we typically apply on S. There's recovery here, and that's it. So they do some basic analysis. I'm not going to look too much at this. And they look at an incidence function, a general incidence, okay? Because the way it's written, it's f of si times I. So this is like f of si times I is beta times ip sq, which we have seen already in the part about incidence functions. And what they do when they study this model, they establish conditions which lead to the existence of a Hopf bifurcation. And looking at p equals 2 and q equals 1 in that incidence function, after some transformations, they get this system here. Not going to spend a lot of time on this. I'll just point out the type of behavior that they observed for this. They observed for this system in the IR plane. So these are much more esoteric behaviors than we have seen before. Okay, so with very little modification to things. So here we have this generic birth rate, the incidence function that is a general incidence function. Under these conditions, they obtain some more complicated dynamics than we have seen so far. Okay, you can see some periodic orbits there, some quite complicated behaviors. Let me, the last of those oddities that I wanted to illustrate was the case of bistable states, which we could sort of qualify as being an undesired effect of vaccination. So this is a model I wrote with McCluskey and Van den Riesch quite some time ago now, almost 20 years ago. So the idea here was to consider an SIRS model, but here we put vaccination. So when I introduced vaccination, I should move myself out of the way. When I introduced vaccination in another lecture, I took this very simplified approach to vaccination, which was to say that we took some individuals and put them directly into the recovered class. Okay, so here we are going to take a slightly different approach. We're going to say that vaccinated individuals, they are moved into the vaccinated class. But additionally, so there is lots of immunity after being immune, but there is also lots of vaccine immunity. So this is what's called the waning of the vaccine, that theta V here that takes you back into the S class. So first of all, notice that there is vaccination at birth, and 1 minus alpha, the corresponding 1 minus alpha is not vaccinated. But once you're vaccinated, you are vaccinated at that rate phi. If you're susceptible and not vaccinated, you end up vaccinated at the rate phi, but you lose vaccine efficacy at the rate theta V. And when you've lost the protection of the vaccine, you move directly back into the S class. And the second characteristic of a vaccine that we consider is that it's leaky. It's not 100% efficacious, which, I mean, if you followed the vaccination against COVID, you sort of are very aware that the vaccines have different efficacies against different variants, for instance. But here we're assuming that of those that are vaccinated, some get infected by the disease. So if, for instance, this sigma is equal to 1, then this term, well, I mean, except that it's an S and a V here, this term is equal to this term. So that means the probability of being infected is the same for those who are vaccinated and those who are not vaccinated. So sigma equals 1 is 1 minus sigma equals 0, and 1 minus sigma is the efficacy of the vaccine. Likewise, at the other end of the spectrum, if sigma is equal to 0, this arrow does not exist. Okay, which means the vaccine is 100% efficacious. Reality is somewhere in between. You would not vaccinate someone with a 0% efficacious vaccine, and unfortunately, 100% efficacy with vaccines is very, very rarely, never achieved. Okay, but the rest of the model is the same. So what happens with this? So the model looks something like this. We are using proportional incidence. So all of this I have said already, so let me go quickly. We transform it's easy to check that the total population is constant, and therefore we can go to proportions. And here I am not showing the proportions as the little s, etc. I'll just keep the capital letters. Okay, and we can work on SIR and recuperate V from 1 minus this. Okay. When we look at the equilibria, we find the following. We find that there's always a disease-free equilibrium, which looks like this. So some of the population is susceptible, some of the population is vaccinated, and we want to find endemic equilibria. And it's okay, so we have a disease-free equilibrium, but now we want to look at endemic. So when the vaccine is 100% efficacious, then there's at most one endemic. But suppose it's not 100% efficacious. What happens? Well, when you're looking for equilibria, I should move myself out. check that. Oh, I was in the way. When you look for equilibria in this model, what you find is that the non-zero equilibria, the positive equilibria of I, are governed by this quadratic equation. This quadratic has its first coefficient negative, and the other two coefficients are there. So what happens? That's the classic case. If we have, I'm going to get it, if we have a forward bifurcation, so what we've seen so far as being a classic bifurcation. So that's what the polynomial looks like, and you can see that first of all there's no equilibria, no positive equilibria. Okay, so I'm looking for an I positive value of the equilibrium for the number of infected. So there's no equilibrium, and then we get to a point where there is an equilibrium, and then that equilibrium increases, which if we transcribe on an R vaccination, I didn't write what RVax was, but it's easy to compute. If I transcribe in an RVac versus I star graph, what I have is the classic setting. When R0 is less than 1, or RVac is less than 1, I have no positive equilibrium. Then when RVac equals 1, there's a branch of positive equilibria that comes in, one branch only, that comes in here, and where I star increases as a function of RVac, okay, which is what you see here on this graph as the star, the point of intersection. It will start out here, and then as this curve moves upward, those points of intersection move to the right. So this is what we have. But there's another situation that arises. In some other regions in parameter space, what we observe is the following. So there is first no equilibria, positive equilibria. Okay. Then at one point this curve becomes tangent, but remember that before it was reaching the P of I equals 0 line here and just increasing with a single point of intersection. Here it reaches, it becomes tangent somewhere here, and then as parameters keep on changing, instead of one positive equilibrium, we find two. Okay, so here you can see this curve intersects twice, and then, okay, so this will happen for some time, and then afterwards, once that part gets over this axis, then again I have only one equilibrium, which takes this form. So what happens is that for values of RVac less than 1, and that's what we observe, we get this situation. So this is where that curve becomes tangent here. And then we have two equilibria for a while, which is what we observe here for a given value of RVac. And come that point where this curve here on the left goes over 0, then we once again have a single equilibrium. Okay. So this is quite different from what we saw before. And okay, we're able to characterize what region this happens in. And so you see that what happens here, this is in terms of sigma, so the efficacy of the vaccine, 1 minus the efficacy of the vaccine, and phi, the vaccination rate. What you see is that there are three different regions. There's a region where there's no endemic equilibrium point, so that's the point where we are here. There's a region where we have a single equilibrium point, which is here. And then there is a region where two equilibria coexist. And along this line is where we have a single equilibrium, so where that curve goes this way. Okay. Oh, we still have some of those. So what we find is two equilibria, potentially. And if we call I star those solutions, then we find that they take this form. So there's an R0 value that you can compute. Let me skip this, it's not really important. We find that the disease-free equilibrium is locally stable when the R0, R5, okay, so this is with vaccination, is less than 1 and unstable when it's larger than 1. Because remember, those stability results, they are local. So when I'm considering the stability of the disease-free equilibrium here, it is locally stable. Okay, the complex, and here it's unstable. So this is exactly the same situation as before. Here the disease-free, which lies here, is unstable. Is stable, and it is unstable here. So that situation is the same, but the difference is that in this case there are also so this is unstable and this is stable. So I think I have it written somewhere here. Yes. So at the smaller I, so that's the smaller part of the bifurcation, the backward bifurcation, the Jacobian matrix has negative trace and positive determinants, so we have a positive eigenvalue. So that lower branch is unstable, and the upper branch is always locally stable. I mean, so we haven't determined it, but we did a lot of numerics, and it seems to always be stable. And I can show you in some cases where it works better. So this is like investigating this local stability of the endemic equilibria as the values the values of theta vary. But we're also able to show, for instance, that the result is actually global in the sense that if those conditions are satisfied, and we investigated, and yes, they are satisfied at times. So if those conditions are satisfied, then all positive semi-trajectories in D, okay, so where this is the constraints on the state variables, they limit to unique equilibrium point. Okay, so you're able to say that you don't know whether you're going here. So you know that you're going to either go here or here. Okay, so you go to an endemic, you go to an equilibrium. Now, let me continue this by showing you a few. So these were my sort of gallery of horrors of things that don't work quite as you expected them, and they exist. The fact that they exist means that sometimes when you're carrying out an analysis, then you might run into one of those. I tend to try to avoid, for instance, backward bifurcations, but sometimes when you write a model, you end up with a backward bifurcation. So you just have to be aware that the situation can be a bit more complicated than the classic situation. Now, let's look at a few models, and they're not all so recent, but I selected 4 HIV models and 4 malaria models, just to give an illustration of things we haven't seen yet. So this first model I chose because I found the title of the article very funny. So I read recently that it is if you have a good title, perhaps your papers will be better cited. And interestingly, I had never heard about this paper, which for me is perfect title, but doesn't seem to have picked up so much. So this was written, I'll point out, this is an HIV model that was produced quite early on in the epidemic. Okay, so at the time, not a lot was known about HIV, much less than now. And that's one of the first models that was produced about HIV. So here, the idea was that they considered that data related to sexual encounters were unreliable or ambiguous, and they wanted to illustrate this through a model. The interesting thing here is that the conclusions they drew was that the epidemiology of AIDS was particularly sensitive to to your capacity to assess the sexual or drug-related behavior, and that given that we have very little understanding of this or very little data, then it makes predictions very complicated. So what they had is this nice little group model that I did not know. So they have M groups. So GI is group I with 1 to N, with size Ni, and they have X as the number of people in group I who are seropositive. And then they look at how those different groups, they look at how these different groups interact with one another to give rise to new infections. Okay, so the model takes this form. So it is written in terms of the number of people that are infected, so seropositive, and the size of the groups. Okay, and you can see that this is an interesting contact term. So QIJ is the rate at which uninfected members of J are infected by infected members of G. So from I to J. It's funny. I did not know this model. I find that it's quite interesting, very simple, and quite elegant in a way. Probably one of the reasons why it wasn't picked up so much was that the study was only numerical. Okay, that's an interesting little model. Let me look at a model of Herb Hefcott from 89, so 1 year later. A model for HIV transmission and AIDS. So this one I'll point out. The reason I'm pointing this one out, so I've decided to show a few examples of a variety of models. So this is a much more meaty model in the sense that if you look, there are many different compartments. So monogamous women, monogamous men, homosexual men, bisexual men, female prostitutes, etc. So this is a large model where there are transmission risks between all the groups and etc. And not only that, but then there is also progression to AIDS from HIV plus that is encoded. So this is interesting in the sense that it's a model that tries to incorporate a variety of factors and makes way for counting the population in each one of those compartments. The next model that I wanted to look at is a much more recent model, and it has to do with HIV testing. Okay. So here, the model has, so I have to look at my explanations. They appear in the slide notes. So these are different, the different stages of HIV, okay? And the different stages of being on antiretroviral therapy. Okay, and so people can move from being HIV plus in the first stage to, and I should move myself here. They move from first stage HIV to first stage HIV under antiretroviral therapy and etc. This is slightly reminiscent of a model that we saw from Michael Lee, Jisheng Shuai, and Hongbin Guo, I believe. We saw in an earlier lecture, except that Guo and Shuai had movement in both directions, except that, and here we've only got movement down this in this direction. But that idea is interesting. The study is quite, I mean, it's and one of the reasons I chose this one, maybe let me go back, you'll see that this was published in the Lancet. So this, in a way, for me, is a good illustration of the fact that, you know, like, you can publish mathematical models in a journal like the Lancet. And for, you know, like, my whole point throughout this course has been choose who you want to communicate to. And if you read this model, this paper, you will see that there is a very big effort that is made in terms of presenting the results of mathematics to an audience that is not a mathematical audience. So this paper, I think, is interesting for us as mathematicians to see what is feasible. You can talk to people in other fields, provided you pay attention to the way you explain things. That's a very interesting model in itself. Okay, I should point out. The last model on HIV that I wanted to talk about briefly is one for viral load monitoring and patient tracing in Malawi. And that's also much more, even more recent paper. And so here they have an individual-based model. So we have seen in one lecture, we've seen one lecture about individual-based models, and I didn't say a lot about them, but I wanted to show you an example where an individual-based model is used. I'm not going to go too much into details, but the idea here is they have what is interesting is it's a mix of a deterministic model for transmission and an individual-based model for how disease progresses in individuals. Okay, so they've got a bunch of compartments in the deterministic model that looks at the HIV status, etc., and then they have an ABM to describe how people go through the different stages of HIV. Okay, so I mean, I was trying to collect a few papers that I found interesting. I found that a combination of approaches is always something that's quite appealing because it is thinking outside the box. So it satisfies my think outside the box. You can use a deterministic model for something that it is good for, and then once you're starting to look at the details of what is happening at the individual level, then maybe an ABM is better, and combining the two is a good way to proceed. Let me talk about malaria now. So I selected 4 models for HIV. Let's look at 4 models for malaria. So I'll say this is a super classic model. This one is quite old. It's by Dietz, Molyneux, and Thomas. Dietz and Molyneux are people that did a lot on malaria. And so this is a malaria model that is tested in the African savanna. And it's in the bulletin of the World Health Organization. So that's an interesting place to publish a mathematical model also. I'll point out that it's not easy to publish a mathematical model in the bulletin of the WHO. So they have quite sort of classic, but not too simple, a description of the different stages of people infected with malaria. So they have non-immune negative, incubating, infectious positive, non-infection positive, then that can go back into this loop, or the non-infectious positive can become non-infectious positive recovering fast, and then they are immune negative and so on. So this is a relatively simple model. The interesting thing here I will point out is that this is a difference equation model, not a differential equation model. So this system is discrete in time. They use a 5-day iteration interval for simulations, or otherwise think about 1 day. But this is a type of model that I have not talked about at all in this course, except when I talked about discrete time Markov chains. But these are deterministic versions of this type of model. Okay. What is very interesting in this paper is that they did quite a lot of numerical work and tried to fit some data that they had for example, the biting rates. They had a proportion of people positive, and they used that data that they had, and they had observed and simulated positive proportion. So this is an interesting model because it goes. Remember, it's 74 at the time. A lot of the models were mostly mathematical. This is a very practical application of a mathematical model. So this is quite important, I think, in this case. There was the super classical. Let's look at classic. So this is a paper by Ngoa and Xu. This is a model for malaria. One of the reasons I wanted to show this paper was to show one important characteristic of modern malaria papers. It's this term here that says that the rate of exposure of humans and the rate of exposure of mosquitoes is both divided by the number of humans. So there is, I've included here some of the justification that comes into this. I'll point out that we also, the paper that I was showing earlier, the first paper that I discussed in this lecture, the malaria one, where we had these weird behaviors at R0, well, at the extinction equilibrium, there's also a good description there of the reason why that is. But it's important to bear this in mind. You can mechanistically derive this. And so remember, when I'm looking at a proportional incidence, I'm dividing by n, the total population. Well, if I'm looking at a proportional incidence in a vector host model, I need to divide by the number of hosts, not the number of hosts plus vectors. Okay, that's one interesting thing here. The model then takes this form. And so you have the dynamics for the population of hosts and vectors. You have the SEIR model for hosts and SEI for vectors, because I should move this here, because vectors do not recover from malaria. Their lifespan is not long enough, so they die while infected. Now, a model where there is some sensitivity analysis. So this is a model by Shipnis, who's done a lot on malaria, and Hyman and Cushing, who are done a lot in epidemiology and population dynamics altogether. So this is a more recent paper. So this one looks at an SEIR model for humans and an SEI model for mosquitoes. Okay, one thing that you'll notice here, that you'll notice in some quite a few models of malaria, is that not only are infectious humans infectious to the vectors, but also recovered humans, because you might recover from a sense of being partly immune to malaria, but you are still harboring malaria, and so a mosquito who would come to bite you could get infected. And so what they do here is that they consider two transmission scenarios, high and low transmission regions, and they do a sensitivity analysis. And one of the reasons I wanted to point out this paper is that if you're looking for values for your parameters for malaria models, then that paper is very, very good. So they look at sensitivity. So this is something I haven't really described, but the idea is to look at how, so this is how R0 is influenced by the different parameters in the model. And in both the low transmission and high transmission regimes. And so what you're able to do is gather the parameters that have the most impact on the changes of R0, plus being that it increases the impact, the value of R0, minus being that it decreases the value of R0. Okay, and so then those numbers are ranked in terms of their magnitude. So you want to know which has the largest effect, whether it's a negative or positive effect. And you can see that essentially here, what's interesting is that the first few parameters, it doesn't really matter if you're looking at low or high transmission, but then starting at this point, the number 4 here, then the situation changes and the more influential parameters are not the same in the low transmission and high transmission regions. Now, another thing that I have not covered, and that is would be worth covering in a beefier course on MathEpi, is the optimal control aspects. So here, this is a model about malaria and COVID-19 dynamics. So this is a very recent model. And what they have is they have a malaria model and a COVID model. And as, and I should get out of the way, as many models that involve co-infection, what you can have, and this I didn't treat, and I think it's worth looking at this type of model even without the optimal control aspect, what you can have is infection solely by malaria, solely by COVID, or mixed infection. So if you already have malaria and you contract COVID, then you become an EMC. If you have COVID and you contract malaria, you can become also an EMC. So there will be models that will consider the order in which this happens. This one doesn't make the difference between the two. Actually, I can't remember what the E here stands for. Yep. But essentially, yes. So this is a co-infection model, and you will see there was quite a lot of interest at one point for co-infection models for HIV and TB, for instance, are two diseases that mix, unfortunately, quite well, because the lowered immunicity that you have as an HIV plus patient makes it more likely to catch TB if you're in a TB rich area. And so this is a type of model that you'll see. So there's also, of course, dynamics for the vectors. And again, like many malaria models, Vectors are supposed to be either susceptible, exposed, so latently infected, or infectious. And that's it. And so they look at a variety of issues, but I'll just point out they do something that as I said, I haven't discussed at all in this course, which is use an optimal control problem. So they use two functions, U1 of t, which is the use of personal protection measures to prevent mosquito bites. So for instance, insecticide-treated nets, application of repellents, etc. And they have a second control measure, which is the use of personal protection measures against SARS-CoV-2. Okay. And so what you end up doing when you're looking at an optimal control is that you write a cost functional, this J here, which integrates over, so it can be infinite or a finite time, but integrates so this is going to be how much infection there was, how much infection with COVID, how much infection, double infection. This is have a number of vectors. So if you want to minimize this, okay, and this is the cost of using these prevention measures, okay, and typically they're squared. I'll be a little cheeky and point out that oftentimes people choose this square term officially because the cost comes in quadratically. More practically, if you have a linear term instead of a square here, you would end up with a control function that's bang bang. The control function is much better looking if you have a U. I mean, I'm not sure that's always the reason, but it very much often is. And so the idea is to minimize this this function. And there's a whole theory that's very interesting behind the formulation of optimal control problems and the resolution of optimal controls. As I said, I'm not going to say much about them except that. But it's worth taking a look at. And so what they find is something like this. So this is U1. This is the control, U2, sorry, and U1. The other way around. So this is the use of, this is an optimal control solution. So this is going to be so remember this is, I'm using full, let's say, personal protection until this point, and this is personal protection U2, remember, is against COVID, until this point, and at which point I can start to reduce my use of personal protection, whereas the personal protection against mosquitoes starts to decrease much earlier. Okay. Now, the last slide I want to go over is what I did not talk about. I just said I probably should have talked about optimal control. Graph models that I've barely mentioned when I was talking about agent-based models would warrant an entire course. Immunology is a topic that I've completely left out. So what we call within host models, these are very important, but they're not mentioned here. For me, one of the very important challenges in Math Epi these days is the Immuno Epidemiology, the interface between what happens within your body and the fact that you're connected with other people and you transmit the infection to them. There is work that is being done on that topic, but it's still very much an open field here, and I think there are a lot of good ideas to be explored here. One thing I did not mention either is the evolutionary aspect of viruses, because viruses evolve quite a lot. I mean, I've mentioned variants in COVID. These things happen over time. Some viruses mutate more than others, but there is an evolutionary aspect which the camera overheating again. So there is an evolutionary aspect in virus dynamics, which is very important and that I have not covered at all. And there is so much more. This is a course over close to 20 hours. To make a decent shot at a Math Epi course, we should probably Math Epi with the aspects that I'm considering, so connections to data, connections to simulations, and so on. You'd probably need 60 hours, but I'm not going to do this here. And that's it.